So we basically set up the stage by talking about ketones, aldehydes, and alcohols before. So carbohydrates is one of the classes of biological molecules that consist entirely of three types of atoms: carbon, hydrogen, and oxygen. And the primary role in biology is the source of energy, as well as building blocks for various cellular structures. So carbohydrates, the simplest, well, not the simplest ones, but carbohydrates are produced, for the most part, in plants by the process of photosynthesis. You see the reaction of photosynthesis in the bottom. When carbon dioxide and water are combined, and there's an energy input in the form of sunlight, this process results in the production of glucose and oxygen. Okay? And then you go back into the cell, put some glucose in, add oxygen, and the opposite reaction called respiration occurs. Okay. Carbohydrates are usually classified based on the number of individual building blocks. So, for instance, monosaccharides such as glucose or fructose or galactose or ribose have just, it's one molecule. See this? Basically, you take this monosaccharides, mix them with water, and nothing happens because it's just one building block. But you can take two monosaccharides and put them together, forming a disaccharide. If you'll hydrolyze disaccharide, you're going to see two monos. And polysaccharides can be hydrolyzed, if you add water, into many individual monosaccharide molecules which serve as building blocks initially. All good? So monosaccharides. The ones that are found in nature contain 3 to 7 carbons. Okay? One carbon always carries the aldehyde or keto group, basically carbonyl group. Does that make sense? Good? Now, classification. If a monosaccharide contains a carbonyl group at the end, if it's an aldehyde, it's called an aldose. If it contains carbonyl group in the middle, if it's a ketone, it's called a ketose. Conceivably, aldoses and ketoses have different chemical properties. Now, in addition to carbonyl and or aldehyde group, there are hydroxyl groups that are attached to every other carbon. Okay, does that make sense? So first classification, kind of summarizing, aldose versus ketose. Another classification that we use is the number of carbons. The ones that we can encounter the most contain 3, 4, 5, or 6 carbons, being named respectively triose, tetrose, pentose, and hexose. Now, then you combine these two classifications. So for instance, aldopentose. It's an aldose that contains 5 carbons. Ketohexose. A ketose that contains 6 carbons. Questions? All good? This is pretty straightforward. Okay, so some examples. This is glutaraldehyde. So aldose, triose, right? You combine it, getting aldotriose. 3 O's, aldo, tetrose. Right, ribose, aldo, pentose, 5 carbons. And fructose, keto, hexose, 1, 2, 3, 4, 5, 6. Okay? So, the compound A, how would you classify it? Aldo or keto? Add the end. Aldo. How many carbons? 6. On the right, aldo or keto? Keto. How many carbons? 5. Keto pentose. Does that make sense? Okay. It is quite a bit of a jump to start talking about hyper and hypoglycemia, but whatever. So, okay, I mentioned that before, when chemists start writing about biology. Normal glucose levels are considered to be up to 100. Okay? So, and it's not just glucose is normal blood level of 70 to 90 milligrams per deciliter. The statement demonstrates a lack of understanding of human physiology. If you are about to, do the blood test, if blood is drawn to test your glucose levels, what should you have done before? Yes. The minimum amount of time, there was a study showing that anything over 6 hours does not really change the parameters. So you can fast for 6 hours, you can fast for 16, it's going to be the same result. Okay? But fasting glucose should be up to 100, no more than that. If fasting glucose is between 100 and 125, it is considered to be pre-diabetic. Okay? 125 and above, diabetes. You can see that the parameters are pretty kind of, I wouldn't say random, but what if somebody has 124 or 126, like how important it is. Now, you can also check the ability of the person to tolerate glucose by doing the glucose tolerance test. In people, it is usually administered orally because it's easier. I did glucose tolerance test in mice. I did intraperitoneal injections because making a mouse drink something is kind of pain in the butt, okay? So if you look at the glucose metabolism that is deemed to be normal, it's not quite exactly correct. Because for glucose metabolism to be considered normal, healthy, the return to the pre-test level should occur around 2 hours, maybe a little more. That makes sense? It doesn't happen here. In hyperglycemic individuals, in people with diabetes, this return will occur days, maybe, if it does ever, okay? So the condition in which people have elevated fasting glucose is called diabetes type 2. Does that make sense? So people have hyperglycemia, and there are 2 mechanisms, as you may know. So what they described is diabetes mellitus type 1, when the endocrine gland called pancreas cannot produce insulin. This is an autoimmune disorder, also known as juvenile diabetes, which usually manifests fairly early in life. Now, there is another condition known as type 2 diabetes. You probably have heard of it. This is a very interesting story because when people overconsume carbohydrates, specifically simple sugars, they constantly force the pancreas to produce insulin, and that insulin constantly acts on the cells to remove the glucose out of the blood. Eventually, cells become responsive to insulin. So insulin is produced normally, but cells resistant, right? Type 1 diabetes is treated by insulin. Type 2 diabetes is treated by diet and exercise. Okay, so symptoms, yes, thirst, excessive urination, but something to be proud of, insulin discovery and synthesis generated 2 or 3 Nobel Prizes. One of those Nobel Prizes went to scientists who were working at Case at the time. It is like one of the insulin Nobel Prizes is there, okay? Hypoglycemia, when glucose levels in the blood go super low. They're not supposed to, but they go. Overproduction of insulin by the pancreas demonstrates yet another time that authors have no idea what they're talking about. Pancreas, okay, in an extremely rare case of a pancreatic tumor that specifically contains Langerhans islets, sure, I can see that. For the most part, hypoglycemia results from the exertion or over-administration of insulin in people with type 1 diabetes when they don't dose it properly, okay? So now let's talk about the chemical properties of sugars. So in addition to sugars, the classic aldoses and pentoses, aldoses and ketoses, there are also sugar alcohols, and we're going to chat about them. So remember, we were talking about oxidation of and reduction of aldehyde groups. Okay? So this aldehyde group, which contains how many bonds between the carbon and oxygen? 2, can be oxidized to carboxylic group, which contains how many bonds between the carbon and oxygen? 3, okay, so more bonds. For instance, by the Benedict's reagent, we talked about this reaction in a previous chapter, in the chapter about aldehydes, when copper oxidizes aldehyde to the carboxylic acid, producing brick red precipitate of copper 1 oxide. Okay? So when we name sugar acids, we take the name of the sugar and replace the ending O's with oleic acid. Okay? Sugars that can do it are called reducing sugars. Now, I'm not sure that it's that important, but we're going to talk about it. So, this reaction that we looked at is the oxidation of glucose and aldose. Okay? It is called reducing sugar because, hear me out, it reduces copper 2 to copper 1. Now, fructose does not contain aldehyde group, it contains keto group. Keto group usually is not oxidized, but in case of fructose, there can be a rearrangement which basically converts fructose ketose into glucose aldose. Essentially, these groups are switching places. Am I making sense? If this happens in the Benedict solution, now you have, instead of fructose, you have glucose, and glucose in turn can be oxidized in that solution. Okay? Sounds reasonable? So this is due to rearrangement. So aldehyde group can both be oxidized to the onic acid, or it can be reduced. And reduction of an aldehyde group results in the formation of hydroxyl group, okay? So I just want to remind you very briefly the chain of events that we discussed in the previous chapter when you have an alcohol which can be oxidized into an aldehyde, which can be oxidized into carboxylic acid. Do you follow my train of thought? Now, each of these reactions can be inverted. So reactions from left to right are oxidations. From right to left, they are reductions. So if you look at this reaction here, the molecule of glucose is reduced to an alcohol. Okay? The proper name is glucitol. So OSE, is replaced with ITOL, okay? Glucose, glucitol, but the common name is sorbitol. Okay? So why is that important? Because the derivatives, the sugar alcohols, can be used as sweeteners. So for instance, you can replace glucose with sorbitol. Why would you do that? Why would you create sugar-free products such as drinks? Why?
To help people, so they're low calorie.
Yes.
Help people with diabetes.
Exactly, it's to reduce the caloric load, right? Help people with diabetes. But what's wrong with a chewing gum that contains sugar? Why do you want sugarless chewing gum?
Bad for your teeth.
Exactly. Because if bacteria on your teeth are getting exposed to sugar, they form biofilm on your teeth much more effectively and start to destroy the enamel much more effectively. They cannot do that if they are surrounded by sorbitol rather than glucose. Okay? So, I'm not sure that it's 160, but we're not going to be picky about the numbers. So effectively, when blood goes through your liver, or through your kidneys, glucose is fully filtered from the blood into the urine and then reabsorbed back, taken back, all of it. Normally, urine does not contain any glucose. When people have too much glucose in the bloodstream, some of that glucose that is in urine cannot be reabsorbed, remains in urine, is expelled in urine, okay? The presence of glucose in urine is glucosuria. And which condition do you think, medical condition, which disease, will result in glucosuria? The D word. Diabetes, yes. People with diabetes will have glucosuria. And we can determine it using a simple test like this. I think one of the labs that we're going to do is going to include a fake glucose, fake urine testing. Okay? So if glucose is present, okay, in the presence of the two enzyme glucose oxidase, it will be oxidized to gluconic acid and produce hydrogen peroxide. And hydrogen peroxide will interact with the dye. This reaction will be facilitated by peroxidase, and that will result in the products of different color, different intensity. Okay? The greater the intensity, the more glucose is present in the urine. Okay, sounds good? So I have a question for you. The year is 1905. Physicians already know what diabetes is, and they know that people with diabetes excrete glucose in the urine. What would be the simplest test? And you are working in some, rural area. What is the simplest test for the presence of glucose in the urine? Taste it. That's the simplest. It was done. There is another one which takes a little longer and does not involve tasting.
Leave it out for flies.
Yes, leaving it out for flies. Yes. Very good. So individual monosaccharides can combine, forming disaccharides. And the combination of different monosaccharides produces, well, different disaccharides. If you combine two glucose molecules, you get maltose, the malt sugar, the one that participates, that is formed during fermentation of like beer preparation, with barley and rye and grains. Glucose and galactose combined form lactose, and glucose and fructose combined form sucrose or table sugar. Okay? So we're going to mention what alpha and beta are, but maltose is the malt sugar, which is formed by two glucose molecules. D refers to chirality, which we didn't talk about, unfortunately, but okay. And alpha refers to the orientation of one of the hydroxyl bonds. Okay? Obviously, hydrolysis of starch, and the forms. Okay, why do we call it an alpha glycosidic bond? How is it formed? This is one molecule, this is another. Does that make sense? How do we know that it's alpha? You see this? You see that these OH groups lie below the ring of glucose? Clear? They lie below, it means that they alpha. If we would have hydrogen here and OH group here, it would be beta. Make sense? So this alpha OH interacts with the OH group and the fourth carbon of a different glucose, forming what's called glycosidic bond. In the name of that bond, we illustrate which carbons are connected. 1, 4. All good? And we also illustrate that the first hydroxyl group is in an alpha position. So alpha 1, 4 glycosidic bond. Good? Lactose. Disaccharide. Okay? So in this case, OH group is above the ring of galactose. Can you see that? Which means it is a beta isomer. Okay? So this will be beta 1,4 glycosidic bond. Okay? Does that make sense to you? Sucrose. So beta, alpha, my bad, and beta. Can you see that? So the structure is quite an interesting one. So the glucose is an aldose. So when it circularizes, it forms a 6-member ring. But fructose, when it circularizes, it forms 5-member ring. Okay? So the bond is formed between the alpha OH group of glucose and beta OH group of fructose. In this case, we signify that we have to mention both alphas and betas for both. So it's alpha, beta, 1, 2 numbers of carbons, glycosidic bond. Okay, does that make sense? Good? Good, okay. So this is an illustration of what alpha beta 1, 2 arrow shows the direction of the bond, what forms to what, glycosidic bond. That's table sugar. Sounds good so far? We produce sweeteners because let's say somebody has diabetes. They can't have malt sugar because it's two glucoses, right? The enzymes are going to break them down to glucoses. They can't really use sucrose because there's a glucose as a part of it. They can't drink milk because it's really hard to drink enough milk to have any diabetic issues really, okay? But there will be some glucose getting into the circulation. This is why a lot of people who have diabetes would prefer to eat fermented milk products, like dairy products, like yogurts, in which the amount of lactose is actually decreased compared to milk. Or get rid of them altogether and just use sweeteners. So sweeteners, compared to table sugar, which is considered to have a sweetness of 100. Okay, so it's an interesting conversation that we can have. So if you use, obviously one of them is, so if you look at monosaccharides, galactose is the least sweet, then there's glucose, and then there's fructose. Fructose can be consumed by diabetics without immediate negative effects. So some of the diabetic foods contain fructose instead of sugar. Okay? You can see that lactose and maltose are also not particularly sweet. Okay? And then we start talking about artificial sweeteners. So you can see that xylitol, the product of reduction of xylose, pretty much has the same sweetness as sugar. But then you start modifying them. And you get chemicals that are 20,000 or 20,000 times sweeter than regular sugar. Okay? So the advantage is you don't need much. Okay? So for instance, this is sucralose. And at the first glance you can say, oh, wait, it looks like sucrose actually. Okay? But there are two chlorine atoms, okay? instead of age groups. Making sense? So this small modification results in a different structure compared to sucrose. Now, here's another question that I have. Aspartame. Does it look anywhere near like sucrose that we saw before? It doesn't. So this is the beauty of biochemistry. When you feel sweet taste, it's because the molecules of sugar bind to the receptors on your tongue, and well, you feel sweet taste. But you can synthesize a molecule that will bind to the same receptors, okay? Stimulate them not just in the same way, but in a much better way, resulting in a much stronger response, so you need much less to make it sweet, okay? So basically aspartame consists of two amino acids, aspartate and phenylalanine. It's a methyl ester, okay? Right here. The biggest problem with aspartame is that there's a phenylalanine part. People have a condition, some people have a condition called phenylketonuria. Have you heard about this? Phenylketonuria. I'm going to go up here. No. Hang on. Where's the chewing gum? Here. Okay? So when you start looking at the list, can you more or less see the list? I see aspartame, the one we just talked about. A part of an aspartame is derived from amino acid phenylalanine. So when somebody chews that gum, phenylalanine gets in the circulation. It cannot be properly processed in people with a condition called phenylketonuria, and accumulation of phenylalanine in the blood will lead, in the long-term perspective, to the permanent brain damage, the developmental problems, and so on. I mean, if it's an adult, the person is going to get sicker and sicker. People with phenylketonuria can normally live and develop. They have to be treated constantly. It's a genetic disorder. But they should not consume anything that contains aspartame, for example. Okay? We good so far? Now let's finish it up. So, monosaccharides. What monosaccharides comprise lactose? Galactose. Maltose. Glucose only. And sucrose? Glucose, fructose. Polysaccharides. Many units of monosaccharides. All right? So I'm going to jump right there. So we're going to focus specifically on the polysaccharides that are formed by glucose. Okay? That include, as you can see, amylose, amylopectin, and cellulose. So, amylose and amylopectin together form starch. What is the difference between the two? So, amylose is the linear chain. It's unbranched. On the other hand, amylopectin is a branched molecule. Can you see that? So adjacent glucose residues will be connected for the most part by alpha-1, 4 glycosidic bond, forming these long chains, but the chains are going to be branched by the formation of alpha-1, 6. This is the carbon number 6 glycosidic bond. Okay? Amylopectin in plants and glycogen in humans have fairly similar structure of bonds. Glycogen is also branched. Okay? So, as you can see, amylose represents about 20% of starch. Okay? It's a straight chain polymer. It's called straight chain not because it is just straight molecule. They are coiled in a spiral, but because there's no branching. Okay, sounds good? I want to ask you really quick. So each individual unit that, which monosaccharide makes up amylose and amylopectin? Okay, the answer is right there. Which monosaccharide? Glucose, yes. So if you break down that chain, what individual units, if you hydrolyze that chain, what individual units you're going to have? Glucose. Keep it in mind. We'll get there. Okay? Amylopectin is branched, so two types of bonds, alpha-1, 4 and alpha-1, 6. Okay? Good? Excellent. Starch in water can be hydrolyzed fairly easily, broken down into smaller chunks. We call them dextrins. And then dextrins can be hydrolyzed eventually to maltose and glucose. Another branched polymer of glucose is glycogen. Glycogen is stored in liver and muscle, yes. So when we need more glucose to get in the blood, we can tap into glycogen storage, break it down, and the glucose will be released in the bloodstream. Okay? So very similarly to amylopectin, the units of glucose and glycogen are connected by alpha-1, 4 glycosidic bonds. Basically, glycogen is like human amylopectin. And branching occurs by the formation of alpha-1, 6. Okay, sounds good? Now, here's what I told you to kind of keep in mind about the glucose monomer and stuff. So imagine you have an enzyme that helps breaking down that bond, alpha-1, 4. Okay? So you start breaking down that bond, and what's going to be released? Glucose. Can you name some food that is fairly starchy that you can just buy off the shelf and start eating it?
Corn or something?
I mean, like basically walk in a grocery store, just grab it from the shelf and start consuming it right away. Bread. Bread contains a lot of starch. Your saliva contains a lot of enzyme. It's called amylase. If you will keep a small piece of bread in your mouth long enough, several minutes actually, you will start feeling sweet taste because starch is broken down to individual glucose molecules and you start feeling sweetness. Okay? Make sense? All right. If you will, now here's another question. Imagine that instead of a small piece of bread, I ask you to tear off a small piece of paper, put it in your mouth. Will you feel sweet taste? I mean, I'm not asking you to try, but you won't. The paper is mostly cellulose. Okay? In cellulose, which basically maintains the structure of plants, okay? adjacent glucose molecules are connected not by alpha 1,4, but by beta 1,4 glycosidic bonds. Okay? Now, this statement is a little bit, it can form hydrogen bonds with water, but not as good as starch. It is insoluble in water. It is pretty rigid. And this statement, I would also argue against it because it is more resistant to enzymatic hydrolysis by human enzymes. Okay? In other words, we do not have enzymes that can break down beta 1,4 glycosidic bond. And it's amazing if you think about this. The specificity of an enzyme called amylase. It breaks down the bond between glucose units, that is alpha 1,4, that goes like this, but cannot break down the bond which is beta 1,4. Okay? So for instance, we often refer to cellulose as fiber. One of the most cellulose-rich foods are celery. Huge amount of cellulose. Cellulose comes in, comes out unscathed. So we don't really benefit from cellulose in grass or wood. But if you will feed grass to a cow, it will process it. Does a cow have an enzyme that breaks down beta 1,4? It's not a cow. It's not a cow. Cow contains, cow's stomach, there's some special term.
They have like 4 stomachs.
Yes, it's multi-chamber stomach, which contains a very rich mixture of bacteria and archaea, and these bacteria and archaea, they have enzymes that break down beta 1,4, and then cow receives all the benefits of it. Okay? All right. Kind of stupid. On the left is cotton. So what? Polysaccharide. Cellulose. And the bond is beta 1,4. On the right, starch, and the bond is alpha 1,4. Okay? I mean, obviously amylose and millipectin, alpha 1,16 and millipectin, but we talked about it. Questions, folks? All good?